Have a nice day to all my viewers. After a long time, today we are going to discuss about another important topic of molecular biology, that is about the translation process in eukaryotes. As we know, the translation is a part of central dogma during which the mRNA converted into cytoplasm, mRNA converted into protein. In eukaryotes, it takes place in cytoplasm. Okay. After some modification in mRNA, the cytoplasmic events stimulate the process of translation. Okay. So, for the purpose of translation, what are actually required? We need for translation, it occurs in cytoplasm first. It occurs in the cytoplasm after transcription of DNA into mRNA. mRNA enter into cytoplasm. Except mRNA, it requires ATS ribosome. This is mRNA, tRNA, transpo-RNA. Except this, it also requires initiation factor, elongation factor, initiation and elongation factors. Then after elongation of protein chain, there must be a termination factor. They are called as release factors. Releasing factors. Okay. Releasing factors. Then enzyme, which is most essential, that is peptidyl transferase. Peptidyl transferase. Then there is, except peptidyl transferase, we also require the source of energy, that is GTP or ATP. Okay, these are the basic necessity to carry out translation process in cytoplasm. Okay, so the eukaryotic translation is a complicated process, because like prokaryotic translation, it never happened. So let's discuss about the detailed process of translation in eukaryotes. Okay, so let's see. So in eukaryotes, the mechanism is similar: initiation, elongation, termination. The process exists similar in prokaryotes and eukaryotes, but in eukaryotes, the mRNA is monocystonic. We know it. Prokaryotic mRNA is polycystonic. Always it produces a single type of protein. In the messenger RNA of prokaryotic mRNA, there is a Sine-Delgarno sequence. What is that? The Sine-Delgarno sequence AGGAG, which became similar with the rRNA segment of 70S ribosome. But here there is no Sine-Delgarno. This is only in prokaryotes. It is not found in eukaryotes. In case of eukaryotic mRNA, what happens? What is the sequence? We have Kozak sequence, which is the binding site of smaller subunit of ribosome. Okay, the sequence is Kozak sequence. Okay, this is Sine-Delgarno, but here we are taking it Kozak sequence. The Kozak sequence is responsible for binding of 40S subunits. 40S subunit of ribosome. Okay. So 40S ribosome, binds before the initiation codon AUG. This is a prime difference between prokaryotic and eukaryotic translation. If we go for the process of initiation first, what is the difference? There are 9 initiation factors. We have to first discuss about the role of all 9 initiation factors, what actually they did. Okay, so come for initiation process. So the initiation is carried out by 9 initiation factors, but in prokaryotes it was 3. Okay, so 9 factors for initiation. Okay, so what are these eukaryotic factors for initiation? Okay, 9 factors. The first factor is eukaryotic initiation factor 2. Eukaryotic initiation factor 2. EIF we are taking for eukaryotes. Eukaryotic initiation factor, the full form. It facilitates the process of binding of formyl methionine tRNA with 40S. Facilitated binding tRNA to 40S subunits of ribosome. Prime event. EIF to V and EIF to 3. Okay. Initiation factor, eukaryotic initiation factor are 2V and 3. 2V, eukaryotic initiation factor 3. These are the factors which are similar in their function. It binds with 40S. Binds with 40S subunit. Okay. Then eukaryotic initiation factor 4A. Okay. Eukaryotic initiation factor 4A. Fourth factor. What it does? It removes secondary structure in mRNA to promote the 40S subunit binding. Suppose sometimes we see there is a coiling or any type of obstruction in the linear sequence of mRNA. So it makes the mRNA sequence completely linear. Okay, that's why the 40S can easily bind with it. So it is checked by binding of 40S and make mRNA uncoiled. Okay, make mRNA suitable for it. Binding. Clear? Then eukaryotic initiation factor 4B. What does it do? Eukaryotic initiation factor 4B. The role of this factor, it helps in finding the start codon of mRNA, that is AUG by 40. It helps in finding AUG to 40S tRNA. They already get assembled one smaller unit of ribosome with tRNA. They get assembled, and this factor help in finding the initiation codon for them. Okay. Then eukaryotic initiation factor 4E. Okay. Eukaryotic initiation factor 4E. What it does? It binds to 5' sequence, cap sequence. Binds to 5' cap. Eukaryotic. Capping became essential to prevent degradation. Okay. Like that, eukaryotic initiation factor 4G. Okay. it binds to poly-A-tail. It binds with poly-A-tail. Then eukaryotic initiation factor of 4E and 4G, they bind to either side, two sides of mRNA. Eukaryotic initiation factor 5, it promotes binding of 40S and 60S subunit. Eukaryotic initiation factor 5, binding of 40S and 60S at this hydrogen. So these are the basic factors. These factors are responsible for different type of function. Okay. So what happens really after that? After the process of initiation, what happens? At first, the mRNA, this is the mRNA, 40S subunit binds to mRNA before AUG at Kozak sequence. Suppose this is the 40S subunit bound. Okay. After that, tRNA bind to 40S subunit. Here, tRNA. Then tRNA binds with it. This is tRNA. Okay. The complex is formed by 40S and tRNA. Okay. They search for the start codon AUG. Okay. Then the start codon is searched. Now, after searching, 60S binds, 60S subunit binds here. Okay. So now the complete complex is formed. AUG, initiator tRNA, these are present in P site. Okay. So when the larger subunit binds here, this is P site, this is E site, this is A, P, E site. The tRNA complex binds to P site. Okay. The A site is free. The next tRNA binds to A site. Obviously, it is free. Okay. Bond formation takes place by enzyme peptidyl transferase. So the elongation started. After that, it elongates. After formation of complex, the total complex started moving on it. The complex started moving on it. So at the P site, when A site is occupied by another tRNA, the complex move. Okay. tRNA gradually get added to the A site. Suppose here, this is one codon. Another tRNA comes here. Already there is a tRNA here with a short amino acid sequence, protein sequence. Okay. So gradually the enzyme, like prokaryotes, peptidyl transferase, remove the sequence from the P site and attached with it here. Then it move forward. After moving forward, what happened? The tRNA of A come to P, the tRNA of P come to E, the tRNA of E goes out one step back. All the tRNA move one step back, and the tRNA which is present at the P side, it has the sequence, protein sequence. Suppose another tRNA came to which side? To A side with one amino acid. Immediately enzyme, what it does? Remove the chain and attach here. Chain attached here, so that no protein in the P. move to P side. Okay, move to P side, and this chain to E side, E to out, one back. So another new tRNA comes here. So the elongation is carried out by this process. Okay. So after elongation, there is termination. So this elongation, after elongation, termination arises. But the elongation factors are there also, which carries the process of elongation. That is eukaryotic elongation factor 1 alpha, eukaryotic elongation factor eukaryotic elongation factor 1 alpha. It helps in delivering amino acid tRNA. It helps in deliver tRNA coupled with amino acid. Okay. Eukaryotic elongation factor 1 beta, release of EF2 and 1 alpha. Eukaryotic elongation factor 1 beta, release of tRNA and 1 alpha. Okay. Then eukaryotic elongation factor 2, it helps in translocation. That is movement. It's carried out by eukaryotic elongation factor 2. So after elongation, we will go for the process of termination. What happens here in termination? In the process of termination, there is a single releasing factor, termination. Okay, the termination is due to single releasing factor, that is eukaryotic releasing factor 1, ERF1, eukaryotic releasing factor 1. It recognize all the three termination codons. Okay, termination codon UAA, UAG, UGA, everything is recognized by this termination factor. Okay. So after recognition by it, the eukaryotic releasing factor 3 stimulate the whole termination process. Eukaryotic releasing factor 3, it help in termination stimulated by eukaryotic releasing factor 3. Okay, then the ribosomes released, the protein chain released, the enzymes became free, the whole process became completed. Okay, so this is all about the process of eukaryotic translation. Okay, if you have any doubt, you can inbox. Okay, thank you all again.